Hello, and welcome to the National Association of Plant Breeders webinar series, The Science of Selections. My name is Araby Belcher, and I will be serving as your host for the webinar. Today's presentation will be given by Dr. Mitch McGrath from the USDA Agricultural Research Service and the Department of Plant and Microbial Sciences at Michigan State University. Dr. McGrath has responsibilities for sugar beet genetics that range from the nucleotide to the field. Today, Dr. McGrath will present a broad perspective of beet breeding and genetics in his presentation, Sugar Beet Genetics, Genomics, and Germplasm Enhancement, or Everything You Didn't Know You Could Know About Beets. I'm going to switch control of the screen over to our speaker, Dr. McGrath.
So, what I'd like to talk about is sugar beet genetics, genomics, and germplasm enhancement. So I'll give you a little bit of an outline of the talk. I'll talk a bit about the industry. I'll talk a few slides of growing beets. I'll look at the crop and its relatives. I'll look at some of the molecular diversity that might be available. Walk you through some breeding systems and some selection. We'll look at some phenotypes. And then we'll transition a little bit to markers. I'll take a brief digression into a little bit of philosophy that you might not have considered using markers. And then I want to get a little bit into the genomics and transcriptomics, and then finish on the state of the art. So in front of you, hopefully you see a pile of sugar beets. And one thing you'll notice is that there are many of them and that they are pretty dirty. So this is a root crop. Every week that the beet remains in the field, it puts on about 1 ton or more of mass. So it behooves the growers to plant early and harvest late. So in addition to being dirty, it's also usually pretty cold. Now those beets will be stored in piles for as long as it takes to process them all. So usually in Michigan anyway, it's November, the harvest, and then the processing campaign isn't done until usually beginning of March. So it is an industrial crop. And if you take the sugar beet from where we just saw it, and I'll be using the pointer a lot, so hopefully I won't be too aggressive with it. But you can see that the beets will go through various stations, so sampling, cleaning, slicing. It goes into a diffuser where the sucrose is actually dissolved out of the beets. These slices are very much like potato chips. Okay, then there's a carbonation step that actually removes some of the proteins and some of the small clay particles, so the purification, filtering, and then it goes through a series of evaporation steps to make what they call thick juice. Okay, and this thick juice is very much like molasses. And after that point, you, in essence, make a supersaturated solution of this molasses, and you inject a little bit of powdered sugar, and you can control the actual size of the crystals that you are going to precipitate. That goes through a series of centrifuges that the molasses passes through. It holds the crystals, and then these crystals are dried, stored, and packed. So the sugar products then would be only one of many possible uses for the beet. And this is a pretty good slide just because it shows a range of those uses, but there are still others. Okay, so as far as the world goes, this is mainly a European crop. So you can see all across Europe, especially France and Germany, are the major growing regions. There are small dots in China, northern China, and then in the U.S. I'll get back into that a little bit, and then in Chile. So those are the major world growing regions. And then in the U.S., as far as 2014, this would be the entire sweetener industry as far as the American Sugar Alliance is considered. And it's roughly about 8 million tons dry weight of sucrose. And the other major sweetener is high fructose corn syrup. I'm not going to talk about that, but of course that comes from corn or maize. And that's all in the yellow. 45% of the sucrose then is coming from cane, okay, and this is, of course, a tropical crop, some in Hawaii, Texas, Louisiana, largest part, and Florida. But the beets are in green then. Here we are in Michigan, okay, Minnesota, et cetera, et cetera. If you look in California here in the Central Valley, I'm going to contrast this a little bit with 2007 in the next slide. So in California in 2007, you actually see a few spots in here. So that used to be a very big growing region, and it actually is being converted now. A new factory has been built for energy beets. Okay, the majority of beets in California are grown in the Imperial Valley. This is a winter crop. In the rest of the U.S., it's a summer crop. And each little blue dot represents 1,500 acres or so. So in total, U.S. 125 million acres, and a worth of about 1.1 and a quarter billion dollars. Okay, so I've been doing this sugar beet research for about 20 years now, and there is a long history of sugar beets at Michigan State. A lot of that has been under the auspices of the USDA. And so this is our farm, former farm, actually, up in Saginaw, Michigan, or Swan Creek, Michigan, if anybody's familiar with this area. And we do very many of the operations that a grower would do: planting, of course. This is the harvester. The beets get topped before they get harvested. This would be a bird's eye view of the farm itself. This particular range down here is where sugar beets are growing, or our plots are growing. It's not a very large area. Some of the other things that are growing on this farm and also in rotation would be dry beans. This almost looks like it's been harvested already. Wheat. We have soybeans. We have corn in rotation. And an occasional crazy crop. Most of what we do needs to be hand harvested, but moved by hand. And I think I calculated early on that I moved 3 tons of beets every year by hand. And I think my back has definitely taken a hit for that. So we're moving much more to pallets and bins. In this particular panel, you can see the folks inoculating, in this case with a pathogen called Rhizoctonia solani. And here's sort of an attempt that we have to use near infrared to scan for sucrose content on the harvester so we can do our selections and not necessarily have to move all those beets. Okay, so if we get into looking at the crop itself, here we have a fairly mature stand of sugar beets somebody was kind enough to dig away. Now this is the taproot. It is a taproot. And those feeder roots can actually penetrate down and follow the water table down to about anywhere between 6 and 10 feet. So it's a very resilient crop in that sense, as long as those feeder roots can acquire the water. As far as the composition of the sugar beet root goes, we have about 75% water and 25% dry matter. Okay, and of that 25% dry matter, about 17% would be sucrose, 7.5% non-sucrose, 5% of that's insoluble, 2.5% is soluble, and we have mainly nitrogenous materials and also some minerals, and these actually interfere with sucrose extraction in the factory. So what we're looking at is the yield of sucrose is equal to the root yield per unit area and the percent sucrose. And that's minus whatever loss to molasses. Sucrose in the physiological situation is, or in solution, is a cation. Actually, it's anionic, I'm sorry. It's got a net negative charge. So these losses to molasses are due to net positive charges, potassium, sodium, and various nitrogenous compounds such as glutamine. Okay, but if we look at over time, so at the bottom we have a list of weeks after emergence, okay, up to 20 weeks. The growers will actually go to 25 weeks or maybe even longer in the Imperial Valley or in other areas of the world where they can grow beets during the winter. They obviously have a longer growing season. But the kinetics then of sucrose accumulation over time are indicated here for different varieties. So red beet, you can see, does not accumulate that much sucrose. And then we can resolve differerences in varieties that actually we've been working with these. These are kind of our lab rat type sugar beets. And what I want to point out here is this line is sort of an asymptote. So sucrose will accumulate to some point where it becomes physiological detrimental to accumulate more sucrose after time. So this is a biennial crop. So as long as it's not induced to flower, it will remain vegetative. So this timeline could actually go indefinitely. So if you look at yield accumulation, though, over time, it's actually quite different. So it just continues to accumulate yield as you go along. Red beets, in this particular instance, are well beyond their vegetable harvesting phase, which probably would be around 14 to 16 weeks, maybe even sooner. And of course, this does not include the leafy beet that you might find in salad greens, which might be harvested at 3 or 4 or even earlier. And just to reinforce that water content has a huge influence on sugar content and yield. So here's a plot of the yield of water versus the yield of sucrose. And you can see that it's got a very, very high correlation. And these are 3-year averages. And the percent of water and the percent of sucrose expressed as fresh weight also has a very high correlation. And this is actually from some F3 families that we have created. And there's no correlation whatsoever between the yield and the sucrose content. So here's some germplasm enhancement data. So we don't actually release varieties. The varieties that we, or the germplasm we develop goes to the sugar beet breeders for the commercial. So here down at the bottom we have a series of commercial entries, and really the goal of this experiment in part was to look at the performance of maybe my entries and versus the commercial entries, and try and get a snapshot of where we are relative to what the commercial or the growers are actually growing. So emergence, you can see one thing here is atrocious across the board. So any seed that gets sold has to be 90+% germination under standard conditions, and in the field, we always get a hit. If we look at the tons per acre, this would be the yield then. we're not so far off of maybe what the commercial are doing, so that's kind of nice. Here in this column is the sucrose as a percentage of fresh weight. Typically the commercials are going to be a little higher in that respect, so we lag behind the commercial entries. So, and then one of the features that the companies, the sugar factories, like to consider is how much sucrose are they getting per acre. And this is really sort of the bottom line. And you can see there's a lot of variation, and these are probably the better lines that we have out. But again, they're not that far off. And it's got to be remembered that these are all hybrids in the commercial sector, while these are just open pollinated materials intended to be included as in hybrids. So percent dry matter and percent water are related very simply in the sense that dry matter and water content equal 1 in total. So this is actually sorted on percent water. So in this case, lower water is equivalent to higher dry matter. And you can see that in general, the commercial materials are slightly lower in water content. Okay. From a standpoint of the percent sucrose as a percentage of the dry matter, okay, it's an entirely different story. And we're not quite sure why that might be. Now, whether these are significantly different or not remains to be seen. But we think that's an interesting way to look at sucrose accumulation as we go forward. Okay, so some of the disease scores are here. These first three, Aphanomyces, Rhizoctonia, and Fusarium, all cause both seedling damping off and more chronic root rot symptoms. And if we just kind of look at the resistant check and susceptible checks here, you can see we're, in our particular area, historically has had a problem with the Aphanomyces. We're seeing Rhizoctonia come on stronger, so you can see the susceptible check here. But there's not too many of these guys that are in our breeding program that are lower than that susceptible check. So that tells us we have some work to do there. Fusarium, we look, pretty good. And this last one is probably the most serious problem in the world, and that's Rhizomania. And Rhizomania resistance is needed for all regions. So luckily it's not that bad of a problem in Michigan. So we haven't concentrated on it, but if our germplasm is to be used in a larger context, we need to pay attention to that. So we've measured that. So it's simply inherited, to tell you the truth, as an RZ gene. Okay, so if I look at the crop and its relatives, we have leafy beets or the chards grown for their obviously leaves, and also the petioles. We have a crop that suffers a little bit from different names. So it's alternately red table garden or beetroot or vegetable beet. Fodder beets are another crop, and then we have the sugar beets, and now we're moving into the energy beets. So this is just taken from some commercial advertising. But basically 6 pounds of beets will give you 1 pound of sugar. Pigments in this family are betalains. They're not anthocyanins, and they're controlled by 2 different genes. So a white beet like this is going to be doubly recessive for the R and the Y locus. You can see sugar beet, table beet, and fodder beet have a certain shape. They've been obviously selected for their root characters. Chard, on the other hand, this is a chard root. These are called sprangles or fangs. It depends where you're at. But this is an unselected character. So basically, if you're crossing one of the root types with one of the leaf types, you don't get a good root and a good leaf. You get a poor leaf and a bad root. So that's the wild type state. So if we look at the evolution of sugar beets and beets in general, you have to start off at the wild beet down at the bottom of the panel here. This is Beta vulgaris. Most of our understanding comes from the Mediterranean coasts, but there's thought to be in Western Asia, which is difficult to collect in right now, perhaps origin of the root, large root character itself. But you can follow this through at your leisure at some time, where we have diversification and also reticulation of the various types, that ultimately result in sugar beet, and we have various flavors of sugar beet. This would be Z for Zucker in German, E for yield in German, and an intermediate type. Spinach beet, those would be the chards. Fodder beets are I wouldn't necessarily agree with their classification here. We have the garden beets here. But it's thought that there's an actual variety that gave rise to, of fodder beet that gave rise to the sugar beet, and this is the white Silesian beet. So if we look at, this is one picture of unrooted tree of genetic diversity. Here we have a complex, a tree that includes Beta maritima, or subspecies maritima, and Beta vulgaris, subspecies vulgaris, the cultivated types. So very, very tightly clustered together. So these are all freely intercrossable. We have Adenensis, which is somewhat crossable, macrocarpa. Then we have another group, or another tribe, Corolla nanae group, and then the procumbens group that really has recently been a new genus. And then the outgroup is Chenopodium alba. And to give you a picture of the Beta vulgaris subspecies maritima, it often grows within 10 meters of mean sea level. So here on the coast of England, south coast of England, we have 3 plants of Beta maritima. And here's Professor Biancardi overlooking a Beta maritima plant down here in the Po River Valley Delta. So this is off, well, the red line indicates probably where you'd find Beta maritima, and again close to the ocean, and likely over in here where it has not been collected as frequently, we might be able to find some other types. So if we look at AFLPs back in the day, amplified fragment length polymorphisms, you can see that from this particular graphic that the sugar beets form a distinct clade from and these are just the sea beet, which is another name for the wild beet. And these are just populations in the Adriatic Sea. So the average genetic diversity between Beta maritima populations is much, much greater than sugar beet cultivars. So of course that raises a possibility that maybe in the sugar beet would benefit from integration of characters from the wild beet. And indeed that has happened, and has been a rich source of genetic characters, particularly for disease traits. So here's sort of a graphic, complicated graphic of the evolution of beets within the U.S. Of course, this was introduced probably into the U.S. as early as the 1850s, but really not until the 1870s and maybe even the 1890s. And through World War I, all of the seed that was planted in the United States was obtained from Germany. So it was really up until World War I before we had breeding in the United States. And these two varieties, Kleine and RNG old type, are thought to be the originators of the U.S. germplasm, at least in these various sites. So the USDA has maintained stations in Salinas, California, Salt Lake City at the time, Fort Collins, Colorado, East Lansing, Michigan. Okay, so this particular graphic has looked at 12 plants per accession, and we scored rapid alleles in this particular case. And one thing you can see that the genetic diversity is slightly lower in the cultivated types versus the wild types. But what we can also see with some other metrics is that within population heterozygosity has decreased, but only with respect to the local area. So, for example, those types that came in from Europe that started off in the midsection of the U.S. have a different genetic profile than the ones in the eastern U.S. And this somewhat reflects the disease pressures that were evident then, and they're still evident now. Okay, so a nice idealized picture of a sugar beet. It wasn't until 1747 that beet crystals were demonstrated to be the same as crystals from sugar cane. And the first sugar beet, that white Silesian sugar beet or beet, was first selected from fodder beet in the late 1700s. And by the 1830s, the breeding house of Vilmorin had selected high sugar by mother root selection. And this is probably the first demonstration of progeny testing. So that's something of note for sugar beets. What I wanted to do is shift gears a little bit to the flowering. And since this is a breeding seminar, I'll talk to you a little bit about the floral structures. So it's an inflorescence. The seeds are born in axils, of the leaf axils. Here's a full flower. You can see there's no petals. And then as the seed matures, it dries down. That seed material becomes then the formation forms the fruit coat. And the actual seed is embedded within this fruit, so the seed doesn't release from the flower ever. And then this is a true seed. So if we look at what we do for flowering in the greenhouse, it's a crop that requires vernalization. So usually this is at 5 degrees C, and we do that during the fall. There's a period of bolting. Now, I'll just introduce the genetics of bolting, and these genes have been recently cloned, so you can go and look at that in more depth. But basically, any biennial is recessive, double recessive for this bolting gene. It's a single gene. And that usually occurs between January and February, flowering actually when these flowers open, and pollen is shed March to April. So we're right in the middle of our greenhouse season, and then processing and harvesting of the seed will occur. So two important features, or this important feature, is a characteristic of every hybrid grown in the U.S. and in Europe and many other places. And that's whether these seeds or flowers are born singly or multiply. So multiply, this is the ancestral state, and there's been a mutation to give only one seed per axil, okay? And that's also a recessive. And you could imagine that it's beneficial in the wild state to have multiple seeds per inflorescence. It gives a better chance for the population to survive. As far as beet breeding goes, it's really pollination control. And you can see right here how small these pollen grains are. So they're windblown. They're also pretty allergenic. So anybody who's going to work with beet breeding is probably going to sneeze or sometimes even break out in a rash. So controlling the pollen is done in many times by male sterility. So you can control who gets the pollen. So here's a fertile flower, and this would have maybe a normal cytoplasm, we'd call. And I just want to contrast that really with the cytoplasmic male sterility. So this, in a sterile cytoplasm, the O-type CMS, or if you're a red beet breeder, this would be an AB type of CMS system, requires 3 genes in a recessive state. Okay, X and Z are usually thought to be the way that we usually talk about them, but 2 genes are linked. So probably Y and Z are linked, but I'm not exactly sure of that. That hasn't been elucidated. But if you work out the genetics, roughly takes about 100 plants in order to get some probability of recovering a plant that is doubly recessive in generating your maintainer lines. Okay, so your maintainer lines are going to be doubly recessive or triply recessive at these CMS restorer genes, but in a normal cytoplasm, and you get the fertility. Another means is a nuclear male sterile, and these are much more easy to distinguish because they're black and shrunken. Because of these 2 or 3 genes, this is almost behaving more like a quantitative trait. Okay, so then here's the seed actually as we harvest it. Here's a multi-germ seed up on the left, and here's a monogerm seed. Okay, taken face on. So if we were to slice this monogerm seed in half, we would get a cross-section of the seed. Now we have on the outside then the remains of that flower, and then here we have sort of an ovary cap. You can see the radical here. You can actually see a dimorphism in the cotyledons. And one of the interesting parts, in addition to the color being not an anthocyanin, is that beets contain what's known as perisperm. It's functionally equivalent to the endosperm that you might be more familiar with, but this is maternally derived. And it's sort of at the edges here of this ovary cap, you can see that it's fairly amorphous, except right here there's a zone of weakness. This particular spile of cells then could be visible here and here. And this is where the radical protrudes, forces the cap off, and then the seed can germinate after imbibition. Okay, as far as commercial hybrid seed production, I hope you can see this. You might have to stand back a little bit. But you can see that there's 2 rows of a pollinator. So this is going to be producing pollen, of course. And then there's 6 rows of a cytoplasmic male sterile. So these could be transplanted. These could be direct seeded. And what we have then, it would be the grower going in and just harvesting the seed from the CMS parents. This predominantly occurs in Europe in the southern portions, which is a hard place to go to because it's right along the Mediterranean, with beaches close by, tough duty. And in the U.S., this is predominantly would be produced in Oregon. So experimental hybrid seed production follows a similar sort of pattern, except here you'd have a single row of a pollen parent, often multi-germ, and then you'd plant different plots of CMS seed parents. Okay, then you could make your test hybrids and evaluate seed from these populations. As far as experimental seed production, about what we do here, We do open pollinated seed production, and we can thresh it at times, so we can grow some fairly large plots. It's possible to do sibling and pair crosses, and here each one of these plastic tents contains a pollinator and a CMS, or maybe two pollinators you want to intercross. So you get some sib mating, you get some pairs crossing, and this all relies then on a complex self-incompatibility system if you're trying to do crosses. Unless it's very difficult to hand emasculate those, but some folks can do that very well. We do use cell fertility. I'll get back to that a little bit. In making recombinant inbred lines. So this is a picture of the greenhouse this time of year, full of pots of beets with bags over them to control the pollen. And you can also put bags over the pollen as they flower in the field. In the field, we'd need to transplant these back out to the field. And all these plants had been grown in the field the previous season. So they had to be harvested and brought back and vernalized and potted up. Okay, so breeding in open pollinated crops is a different bear than breeding in self-pollinated crops. And the game is to manipulate gene frequency, where the gene functions are known particularly. And of course, that's where we'd like to move to is know what all these genes might be doing, so we could simultaneously select for them all. But it's a random process. So an original population, let's say we just want to remove those red balls. At random, we remove, some number of balls with replacement. And this is a random process. And slowly we get to the point where we've removed all of the red balls. Okay, so as far as beet goes, then we have this idea of the mother roots. Those would be the ones that we took out of the field, and just doing mass selection with or without progeny testing. We could do sib mating, and of course we could do this inbreeding via the dominant self-fertility allele. So this is very nice that it's dominant, and then it suppresses self-incompatibility and acts as if it's a single gene, but we don't know whether it's related directly to the self-incompatibility mechanism or if it's a suppressor of that self-incompatibility. But hybrids are enforced with the nuclear or cytoplasmic male sterility. So there's basically a degree of inbreeding power, okay, and certainly your greatest inbreeding is going to happen when you self-pollinate. So the predominant diseases of the U.S. growing regions are somewhat related to the USDA breeding stations, who all have responsibilities for breeding for resistance. So in Michigan, like I mentioned earlier, Aphanomyces, Cercospora, Rhizoctonia, and some cyst nematode are all problems. Minnesota is very similar, and you really get a divide between east and west due to this curly top. So we can almost have two different heterotic type groups in the U.S. depending on whether the germplasm originated in the western part of the U.S. or in the eastern part of the U.S. So curly top has been that driver, as well as rhizomania is a lot more important. The viruses in general are important in the western regions versus root rot diseases in the eastern portion. So I'll just show you a couple of pictures of some of the work that we've done. There's mass selection for Cercospora leaf spot resistance. That resulted in this particular trial, we had a very quick burn down. July 27th to August 15th is how the fields actually look. And this is a variety or a germplasm release called EL50. And it serves as our resistant check now. Here's another example then for Rhizoctonia Crown and Root Rot. and also damping off resistance. So in East Lansing, Michigan, this was done on campus then. It was July 7th, and this is a progressive disease, and August 21. But as this disease, as the season progresses, the disease will kill more and more plants. So here, and so here's a selection that we released as SR98 with, and this one actually has damping off resistance, and that damping off resistance also transfers through to the adult form of the disease, which is called crown and root rot. We don't know if they're the same or different mechanisms. So then using self-fertility as a tool, in this case to examine segregation for sucrose content, this is one of our recombinant inbred populations. Here's just measured at the F2 generation of greenhouse-grown plants. But here we have a root, the individual F1 root. Here we have a red beet parent. Here we have a sugar beet parent. And the color indicates, of course, the color of the root. So red being dominant, follows a nice 3-to-1 segregation pattern. But what I'd like to say is that we do get transgressive segregation for sugar content on a dry weight basis. So if you look at those plants in the field, the progeny of those plants in the field, you can see segregation between families or differences between families, and then also within families, and this is going to be the F3 generation, and we call this the MSR population, just in case that comes up again. So we get to the F5, we're starting to inbreed, okay. Here we have these growing in the field. We've got a little processing line up here where we're weighing the beets. We took 5 beets. It should be pretty inbred. The variability should be, due to genetics, should be fairly low at this point. And then here we're scanning with the near infrared for sucrose and water content. So we see rather low within family variation, okay? So these would be 5 of the beets selected for analysis, and you see long beets, you can see round beets. Actually, this particular year, we set up a little bowling pin alley just when one of the machines broke down until it got fixed. But what you can see is also high between family variation. So there's an extreme amount of variability, almost encompassing all of the root types that have been described in beets. Just in this one population, okay, and this is one population is derived from a single individual, F1 individual, that contained one gamete, one pollen grain from a red beet, and one gamete, one ovule from a sugar beet. So the results of all that weighing suggest that there's a lot of environmental variability for root weight. Okay. So it's not a very good character. It very much depends on a good behaving character as far as a geneticist would be concerned. Okay, but sucrose content, there's quite a lot of variation in sucrose content. It's kind of surprising to see less water content variation, but I think that's a matter of the scale that was used here. But it's very tight variances around that. Water content, and of course the dry mass of the dry matter of the biomass is quite variable. So we can start to now think about what's heritable and what's not heritable, and you'd think that would be known for any crop, any trait that you'd be looking at, but it's actually a very simple initial foundational question to ask. And so one way one can do that is to partition all that phenotypic variability across the chromosome landscape here, basically doing QTLs. And I should have actually made 2 different slides for this. What I wanted to get across here was that for these different traits that were measured, so the percent of sucrose as expressed as fresh weight or dry matter content, There is a peak observed at this locus for sugar yield and root weight. Okay, these would be mass. These would be grams per something, tons per acre, mass per unit area. That those are scattered throughout the genome. There's not that many of them in this particular population. But what was striking, though, is this water business starts showing up again. And here we have almost every trait subtended by a water trait. Okay, there's very few water QTO. There's very few of these traditional measures that are not influenced by the water content. Not quite sure what to do with that yet, but raises some interesting questions. So I want to briefly digress a little bit here and look at sort of elements of modern plant improvement in a very simplistic fashion. Of course, you've got populations, okay, and then you do some real breeding based on some phenotypes, and then you take those, selections, and then you make new populations, and you're back and forth and back and forth and back and forth. Okay, so this is sort of the real breeding process, and this markers is in quotations because I, in essence, consider anything a marker that gives you some context and clarity to this process. So our markers can be recently applied to populations, and this would be the inheritance, and we just went through an example of that. And this is more related to the phylogeny, that we can understand a little bit of the phylogeny and a little bit of the inheritance by looking at markers in populations. But one thing that I think is also important to realize is that we can look at phenotypes with markers as well, and this would be the ontogeny of the development of the root or the plant or whatever organism you wanted to look at. Okay, so that's what I want to go through a little bit right now. All right, so I mentioned earlier for emergence as a phenotype. Okay, so seed has 95% germination that's planted in the field, and it averages only 60 beets that are established. So that, in essence, is shown here in this graph in the field, 97, 98, and 1999. You can see a high vigor and a low vigor germplasm. These are statistically significantly different in their emergence, the low vigor, of course, being low. The traditional test, the commercial test for germination potential is done, in essence, in filter paper, wetted filter paper, and there's no significant difference here, and there's high germination. We decided that we would try something different, and we would germinate seedlings or seeds, place seeds in water, and then count them after a certain period of time, see how many germinated. So this is how many germinated in water. And you can see that does reflect what's happening in the field, not necessarily magnitude, but certainly in relative. And then we started adding things to the water until we got to hydrogen peroxide. Okay, so everything germinated in hydrogen peroxide. So then the task was to try and figure out maybe what was happening here. So here again, we're looking at some sort of gene expression between low and high vigor sugar beet seedlings. Okay, and so in this particular instance, 95% of what we looked at did not vary between germplasms or treatments even, and 5% did. And out of these 5%, we were able to look at find one member of a gene family, okay, that showed some differential expression only in the high vigor germplasm and only under stress relative to the low vigor germplasm, and it's not expressed in either filter paper or under hydrogen peroxide. So it turns out this thing is a germin-like protein, which is a putative oxalate oxidase. Now, oxalate oxidase takes oxalic acid, which is plentiful in the beet seed, and makes carbon dioxide and, lo and behold, hydrogen peroxide. Okay, so this is an association that was made that it kind of makes sense, but we don't have any serious proof for that. But here's sort of a model then. You've got your seed here, the radical, then the cotyledons, perisperm. Okay, stress-induced germin-like protein expression or exogenous hydrogen peroxide acts as a vigor signal, an intracellular signal, a secondary messenger. And then this is recognized and transduced, and we think it's through a MAP kinase cascade. That's what the literature would tell us. But these are actually very difficult for people like myself to work on because we've got too many phenotypes that we're looking at and too many plants, which would suggest an activation of gene expression via specific transcription factors, and these are currently unknown. But we do have an induction of the glyoxylate cycle, which is very specific to seed germination and very well characterized in other lipid-utilizing seeds. And this is really a marker. So basically what's happening is this hydrogen peroxide is inducing some substrate by which the plant can use for heterotrophic growth. Okay, so in essence, a vigor gene. Okay, so then if we could then make that more applicable to any trait, we could say we're looking for transcriptomes. Can we use transcriptomes for trait discovery? And this actually accomplishes a number of different things. It allows you potentially to identify the genes as well as the processes that are involved and use those for markers. So just as an example, here we have germinating seedlings, and this is all of the high vigor germplasm, and we haven't contrasted that with the low vigor germplasm. But if we were to characterize then by some sort of name what those genes that are in these wedges were biological processes, then we have, for the germinating seedlings, we have about 26% of them are devoted to catabolic functions that are breaking down molecules to allow for growth, okay? Now, if we get into maybe developing roots, and here's a series of roots that are developing between 3 and 10 weeks, we can see that fraction that are devoted by catabolic or to catabolic processes is reduced by about 10%. So these plants are now phototrophic growth and have to devote less to catabolic processes. So that would be an example whereby these transcriptomes, you could start to see differences that could be related to germplasm and help understand some of the maybe traits that have low heritability, okay, and begin to understand how one might breed for those. Okay, so a couple more slides. We definitely needed a better genome sequence so that we can interrelate genes and phenotypes in a more facile fashion. So Sugarbeet, I haven't told you, is 750 megabases for its genome with a haploid content of 9 chromosomes. So is under 100x, you can actually see the chromosomes. So there's not a whole lot of differentiation. But here's in this particular panel, a phylogenetic relationship between monocots and dicots, and the Caryophyllales, from which Beta vulgaris is a member, forms a different group. There used to be a trichotomy or a polychotomy between the asterids, rosids, and the Caryophyllales, but it's now becoming a little more apparent that the Caryophyllales broke off from the asterids prior to the diversification of the rosids. But these are some sequenced genomes. So Beta vulgaris is the only sequenced Caryophyllales genome that's been published out there. So we can follow the Caryophyllales clade in this particular instance and arrive to the Amarantheaceae which includes beet, spinach, amaranth, quinoa, and many, many weeds. But this whole family, that comprises about 10%, well, about 7% of the plant diversity or the dicot diversity, is characterized by some very strange critters like Venus flytraps and cacti and ice plant, which is commonly seen in roadsides of California freeways, as well as some other things that you'd recognize. Characterized a lot by stress-tolerant species. So just to give you a current status of the beet genome sequence, we basically have about 27 to 30,000 genes in that range. So we think we have good gene space coverage, but we have way too many contigs. So here's some different lines that have been sequenced to some various depths using various ways. This is really quite an interesting challenge to assemble a genome. So this first one, RefBeet, has been published as of last year. Too many contigs. So it's very difficult then to associate a contig with a chromosome. And there's reasons for that. But we're currently pursuing a reference quality genome using next generation sequencing scaffolding libraries that are slightly different, using optical mapping, using a hybrid approach with next-gen sequencing and back libraries pooling, and then also the genetic maps. And what, in essence, we'd like to do is use all this information. In the background, you can see that it's a network. This is an example of a network of beetroot development. So which genes are interacting with other genes and their strengths of their interactions. The biggest problem is, what do we define these particular genes as? So that hydrogen peroxide responsive germin-like protein, if you look in NCBI and do a blast, the top hit will be carnation, a protein from carnation called dead petal protein. Okay, it has absolutely nothing to do with its function. It has entirely to do with its phenotype in carnation. But yet, if you were to annotate your gene as that, you would have lost the ability to recognize that that were an oxalate oxidase and potential vigor gene. So I want to thank you for your attention. I want to thank everybody in the program here, in the Sugarbeet Research Program in East Lansing, to many, 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 many colleagues from around the world. We get our funding from USDA ARS-based funds, and we get direct and in-kind assistance from the member companies of the Beet Sugar Development Foundation, also Michigan Sugar Company, and also Michigan State University. I want to thank Araby and Alice and all the other organizers, and also the presenters as well of these NAPB plant breeding webinars, because this is an outstanding resource, particularly since they don't go away after the presentation is over, which is actually kind of scary. And especially, I want to thank you guys for your time, attention, and interest. So thank you very much.
Okay, great. Well, I guess we can close it up then. And as a reminder, and again, thank Dr. McGrath. As a reminder, a recording of this webinar, as well as all other webinars from this series, will be available at extension.org/plant_breeding_genomics. And again, thank you so much for joining us, Dr. McGrath, and thank you to our audience for coming.